Okay, let's talk a little bit about the genetic material, DNA and RNA. So first we'll look at them both together. There's a little video that I'm going to put on Google Slides for you so you can take a little gander at it and look at DNA versus RNA. But if we look at them both together, they both fit into the category of a nucleic acid. So remember, everything can go into one of four categories: carbs, lipids, proteins, or nucleic acids. Now, they're both made of nucleotides. So way back when in chapter 2, we talked about monomers versus polymers. So monomers being the building blocks, nucleotides, and the polymers being the larger molecules, DNA or RNA. So they're both made of these structures called nucleotides. You can see one here. Okay, so we'll look just at DNA. So this little guy right here. DNA has a nucleotide that's made up of a sugar called deoxyribose sugar. And the only difference between this sugar and the sugar that you're going to see in RNA is that this one is lacking in oxygen. The bases, so these are the nitrogenous bases or the nitrogen bases. These bases, this is your genetic code. The bases are adenine and thymine, and they pair together, and cytosine and guanine. Now, I said that they pair together, and they pair together because DNA is made of two strands. It's referred to as a double helix. You can see it there. All of these little rungs of this little twisted ladder, those are your nitrogen base pairs, and all these little rungs of this ladder, that is your genetic code. That is what makes you you. So as we've been talking about all of these traits and alleles for traits, all that information is right here in the rungs of this ladder. Now RNA made up the same structure, nucleotides. The sugar is called ribose sugar, so it has an extra oxygen. That's what separates the two. It does also have bases. These bases, a little bit different. They're still adenine, but there is no thymine. Instead, there is uracil, and then there's still cytosine and guanine. Now, they won't be pairing together here because RNA is just a single strand. So it's a single-stranded structure. Okay, so let's look here at DNA just a bit. Now again, there's a nice little video here about the structure of DNA and what it does, and I'll put that on Classroom. But DNA itself stands for deoxyribo, that's the sugar, nucleic acid. So this is something that pretty much you're going to encounter wherever. So here's the structure of a nucleotide. A nucleotide is made of a phosphate group, which is this phosphorus and oxygen, connected to, by a covalent bond, this deoxyribose sugar. These two parts make up what's called the backbone of a nucleotide. So if you were looking at this in terms of a ladder, this would be the sides of the ladder. And then the sugar is connected to one of the four nitrogen bases. And the sugar and the nitrogen bases may vary a bit if you're talking about DNA or if you're talking about RNA. But we're in DNA right now, so we're talking about deoxyribose sugar, and we're talking about A's, T's, C's, and G's. Now, I love memory devices, so I'm going to provide you with some memory devices when it comes to the nitrogen-base pairings of your DNA. I also have a little device here for remembering which one is which in terms of a purine or pyrimidine. So here are your four nitrogen bases or your nitrogenous bases. And adenine and guanine are called purines, and cytosine and thymine are called pyrimidines. So this you'll definitely encounter again in higher level science classes. So an easy way to remember which one is which is purag, P-U-R-A-G. So if you say purag, that means that the purines are adenine and guanine. And then that would leave cytosine and thymine for the pyrimidines. Now, that's not my personal memory device. I just snagged that one from some study group that I found on the internet. But the way that I always remembered which one is which, purine, smaller word, but it's the bigger molecule. Pyrimidine, bigger word, but the smaller molecule. And that's an easy way to remember a lot of things. That's an easy way to remember acids and bases, and that's an easy way to remember surface area to volume ratio and the size of a molecule is by using opposite day. Now, when it comes to who pairs with who in DNA, we can use this little gem right here. All turkeys can gobble. So A pairs with T. They're complementary or complementary bases. And then C pairs with G. Now we're going to talk about making copies of DNA. So we know that at certain points in a cell's life, DNA has to be replicated. This process of replication is going to occur before mitosis. It's going to occur before meiosis, during interphase, specifically the S phase of interphase. Now, the process itself is called replication, and the goal is to make two identical copies of the DNA. So here we have the original, and then here we have the two copies. Now, this process is going to occur in the nucleus of a eukaryotic cell, like ourselves, and in the cytoplasm of a prokaryote. And every time this happens, you end up getting a new piece of DNA that has an old strand and a new strand. So this old strand is actually called the parent strand and serves as a template to make the new strand, which is called the complementary strand. Because again, we're talking about complementary bases. So when we go to do this, it's very simple. You just have to remember your nitrogen-based rulings, A with T, C with G. Now, if you look at this, though, you can see some numbers, 5s and 3s and 3s and 5s. So this all has to do with what's considered top and bottom. So the DNA molecule is called anti-parallel. So if you look, one strand is going to be right side up, the other strand is upside down. That's the way it works. Something else about this 5 and this 3, all that has to do with is positioning of the molecule. So which carbon is at the top? Is it the 3 or is it the 5? And that's where you get these numbers from. So you'd probably deal a little bit more with that in biochemistry. So let's replicate this one. So all I have to remember, all turkeys can gobble. So C can gobble. C will go with G. G will go with C. T will pair with A. A will pair with T. A will pair with T. G with C. C with G. G with C. G with C. C with G, and T with A. So it's that easy. Now, what keeps them together, and you can see like these little dashed lines right there. So at this point, they have to be unzipped to separate the two strands, but when you go to form them, these will get longer, they'll connect together. The bonds that hold the two strands of DNA together are called hydrogen bonds. And the hydrogen bonds are what are responsible, or here they are, there, the hydrogen bonds. They're responsible for the twist in the DNA. So here's another one from practice, so you could see what you could do with this one. Remembering all turkeys can gobble. And this should be what you have. C with G, A with T, G with C. And then what we would have in here would be the hydrogen bonds. And replication is that easy. So then this piece of DNA will go on to maybe be in the daughter cell. So if you're making some new skin cells in mitosis, you would need a copy of the DNA. You don't donate, you have to give a copy. Now this process works out pretty well. There is an enzyme DNA polymerase that's responsible for putting the bases together It's also responsible for proofreading. So it's kind of like a couple of jobs there, putting together and make sure that it's putting the correct bases together or the complementary bases together.